Good morning, everybody. Thank you very much for inviting me for this very interesting congress. And today I will talk about the evolution of insecticide resistance in mosquitoes, not only in Aedes, because most of the examples we've got come from the Culex complex and from Anopheles, but whenever possible I will talk about Aedes. So it's going to be quite basic science, but sometime I will show you that basic science can be applied to vector control. So you know now that mosquitoes are resistant to most main chemical insecticide like pyrethroid, carbamates, organophosphate, and cyclodiene. And the main mechanism of resistance are target modification, metabolic resistance, and behavioral resistance. Well, we talk more and more about this last resistance. I will say a few words about that. So the example we've got about behavioral resistance comes from the Anopheles complex. So we've got few examples in Anopheles gambier. So the mosquito are changing their behavior and they go outside for biting to humans. And we've got also two examples in Anopheles funestus. And here we see that they change the timing of biting. So we don't have any example in other kind of mosquitoes. And the question is concerning this resistance, is it heritable? So we don't have any data about that. And what are the genetic basis of this kind of resistance? What will be the evolution in the field of such a resistance? That's very important, of course. And will it be a problem for mosquitoes protection? I mean the mosquito net. And also, does this behavioral resistance exist or will be selected in other vectors like Aedes through other kind of selective pressure, because we don't use the bed nets against Aedes. So that's something we need to study in the future. Concerning the metabolic resistance, I will describe that very briefly. So there is a lot of enzymes which are implicated in metabolic resistance, and with new sequencing generation data, the list is getting more and more longer. Resistance to insecticide in that case is associated to increase quantity of enzyme or increase activity of enzyme. Most of the time it's superfamilies of enzymes, so it's difficult to identify the genes or mutations which are implied in resistance. And so it's very necessary now to genetically and functionally validate the gene and mutation which are implied in this kind of resistance. And also we need to develop diagnostic tests to follow the resistance in the field. And I think the Dr. David will describe much more about this kind of resistance, so I won't talk about metabolic resistance anymore. Concerning target modification resistance, so most of the time it's punctual mutation in the gene coding for insecticide targets. And we've got for the chemical insecticide I described, we've got three main targets. It's the acetylcholinesterase, coded by ACE1 gene. It's receptor GABA, and it's sodium canal targets, so with the KDR gene. All these targets are very constrained proteins, so it means you can't mutate them without, most of the time, completely delete the activity. So there is very few mutations implicated in resistance that were be found. And sometimes we've got synergy between several mutations. And there is molecular diagnostic test for resistance to detect this mutation. These tests are very useful, but I would like to point something here that these tests are useful, but sometimes they may prevent detecting new mutation, because we use all the time the same test to detect the mutation we know. And after that, and I know that because I developed tests in my lab, After that, we don't know if there is some other kind of mutation that are implicated in resistance. So it is important very often to do some survey and to use a bioassay to see if the resistance vary in the time. I won't talk too much about that because we'll have the talk of Dr. Kasai and Dr. Vontas later on. Concerning the evolution of resistance in the field, we know that the mutations which are selected depend on selective pressure and the local history. That will be the case for the KDR leucine to phenylalanine in West Africa and the leucine to serine in East Africa in Anopheles gambiae that were selected because of different pressure and local history. Concerning Q-excipient complex, we found the glycine to serine 119 in most of Culex, Anopheles gambiae, Anopheles albimanus in the world, because it's a larger resistance spectrum we could find. But we found another mutation, the phenylalanine to valine, around the Mediterranean Sea, because it was a better resistance ratio we could find against insecticides that were used especially there. So this is to stress that the environment is also implicated in what will be selected in the field. Also, I would like to point out that it's not always the same mutation depending on the species. You may know the story of the molecular constraints of the codon. So we found that sometimes the glycine 119 codon is what we call immutable. So it's a case for Aedes aegypti and Aedes albopictus. They've got an immutable codon for the position 119. So you can't get the mutation glycine to serine in position 119 in aegypti and albopictus. Or maybe there is some, this mutation is coming to India now because of this molecular constraint. So the main question we ask in my lab is how does resistance evolve in a field? And we know now that it will depend on genes, it will depend on mutation, and also one main factor, it will depend on the impact of resistance on phenotype and fitness of the mosquitoes. And also another factor is the pressure from the environment. So if I go back to the story of my favorite gene, and which is ACE1 gene coding for the acetylcholinesterase. So we've got the susceptible gene with glycine in position 119 and the resistant gene with the serine in position 119. So the serine will block the active sites with the lateral chain that will block the insecticide that goes to the active site. Oh, sorry. But this will also block the natural substrate, and that will decrease the activity of the enzyme. And this will account for a very high fitness cost. So the mosquitoes who are resistant through this mutation in the acetylcholinesterase gene, they suffer the very high fitness cost. And we measure that in the field, and we measure that in the lab with strain sharing the same genetic background on several life history traits in Anopheles gambiae and in Culex pipiens for several life history strains. So, I mean, resistant mosquitoes, males mate less than susceptible mosquitoes, okay? They develop, they take longer time to develop. They die really much more, okay? The fecundity of resistant female is really lower than the fecundity of susceptible female. The resistant mosquitoes are much more predated by predators. So every time we did measure on that, resistant mosquitoes suffer a very high fitness cost. We've seen the same kind of results that were presented by the laboratory of Dr. Martins here in the Aedes aegypti with strains sharing the same genetic background. So in Aedes aegypti with the carrier mutation, the resistant mosquito took longer time to develop. The female laid fewer eggs. And in competition cage, the resistance decreased and the susceptible mosquito wins. Okay? So they also suffer a very high cost. So why do I insist so much about this fitness cost? Why is it so important? Because this is a very important parameter for the evolution in the field. And it is also a very important parameter for vector control. Because if there is no cost, there will be no management of insecticide resistance by rotation or by mosaic management, okay? So we need this cost for resistance. And the more the cost is, and the best it is for management. We measure that. We had the opportunity to measure the cost in vivo in Montpellier area because we followed the resistance in Montpellier, south of France, since more than 40 years now, every year. And there is a European, program that said that the DOP could not be used anymore in 2006, okay? So we measure that resistance frequency since several years, and we decided to study what the stop of the OP will bring to Montpellier area. And that is what we had. What you can see, it's for S1 resistance gene. The decrease goes very fast after the stop of the OP treatment from the vector control. Okay? For esterase gene, which is a metabolic resistance system, after the stop of the OP, we saw the decrease, but then we've got a plateau here, around 40%, that didn't move since then. So it means that the stop of the OP for this very high fitness cost resistance is very efficient here, but here, for this generalist resistant gene, we've got a problem because we still have a selective pressure that selects for esterase genes. And we think, from what we see in Montpellier area, that the selection pressure is coming from agricultural, okay? So it's very important to think about control insecticide resistance and to have a good way for this control, but we need also to think about this other kind of selective pressure. And also concerning the impact of resistance on fitness cost, what is important to understand is that, distinct mutation will have distinct trade-off between resistance and cost that are adapted to distinct environments. So you've got several factors: mutations, impact on fitness, resistance and cost, and impact on environment. So here we've got the acetylcholinesterase again, susceptible resistant. And in Anopheles gambier, we found several kinds of duplication, homogeneous. So it means several copies of the resistance here. So in fact, it's not just GS1 gene which is amplified. It's 200 kb zone with 12 genes, in fact. And we've got heterogeneous duplication, so with one susceptible and one resistant copy here, okay? And all this, you can find that in the field in Anopheles gambier now. And what we found, it's when you've got several copies of this gene, you've got a quantitative advantage, so the mosquitoes are more resistant to insecticide, but also you've got an increase in the costs, metabolic costs, because you've got all these 11 other genes, so the cost is increasing. And this is well adapted to high selective pressure. So the more insecticide you put in the field, the more copy you will get. And when you've got a kind of mosaic or medium selective pressure, you will select for duplicated heterogeneous duplication that behave like heterozygotes. So you have several types of mutation who have different impact on fitness, and they can adapt to different kind of selective pressure in the field. So that will be a problem on vector control, because depending on the selective pressure, you've got duplication that can adapt to several kind of pressure. Okay, so the last thing I would like to discuss here, it's that also we've got problem with influence of insecticide resistance on vectorial capacity. We've also, this was shown on Anopheles gambier, that the resistance is interacting with the vectorial capacity for the parasite Plasmodium falciparum in Anopheles gambier. So there is an interaction between resistance and vectorial capacity for Plasmodium. So it was shown, for both time it was for paratoroid. Sorry, it was paratoroid here? Yeah, paratoroid here and paratoroid here as well. So the question is, will we have the same kind of interaction between ADES, resistance, and virus? I guess yes. And I would say also that we'll have interaction with microbiota, temperature, a lot of different things that will interact. So maybe that would explain why you've got this kind of good competence in this part of Brazil and not in this other one. So that's this kind of research we need to develop. So how the resistance, the temperature, any kind of microbiota can influence the vectorial capacity of mosquitoes. Thank you very much. Hope you will have any question on that. Thank you.